Hi there, welcome to INVODENT sessions. This is Dr. Monica, orthodontist. Today let us try to understand what is glycolysis and what the various steps that are involved in glycolysis. Before going into that huge cycle and taking around, let us first try to understand what is the meaning of the word glycolysis. Glyco means something related to glucose. Lysis means breakdown. So breakdown of glucose to give the energy molecule, that's your ATP and pyruvate molecules, is all about glycolysis. Before we go into the further steps of glycolysis, first let us try to understand the basic unit, that is about the glucose. First let me tell you what is the chemical structure of the glucose. Glucose is composed of C6, H12 and O6. So there are 6 carbon atoms, 12 hydrogen and 6 oxygen atoms. So 1, 2, 3, 4, 5 and 6. So this is your glucose structure. We eat a lot of carbohydrates. The carbohydrates are getting converted into glucose. Now this glucose is stored in the bloodstream. We want this glucose to break down to release the energy by the process of glycolysis. Where does this glycolysis takes place? In the liver. So now we want this glucose which is present in the bloodstream to reach the hepatocytes, those are nothing but your liver cells. How does this glucose enter the hepatocytes? Hepatocytes won't allow macromolecules like glucose to enter into them directly. So there is something known as glucose transporter. This is a carrier molecule which is known as glucose transporter 2. With the help of glucose transporter 2, the glucose in the bloodstream is carried into the liver. There are several types of glucose transporters which are very specific to each and every organ. Glucose transporter 2 is specifically involved in transport of glucose from bloodstream to the liver cells. Now, the glucose is entered into the liver. Now what happens? The glucose gets converted into glucose 6-phosphate. Glucose 6-phosphate. First point is that why does the glucose should convert into glucose 6-phosphate? Because we don't want the glucose which have entered the liver to leave the liver again back. We want the glucose to stay back in the liver to proceed with glycolysis. For that to happen, we need to phosphorylate the glucose. That means we need to add a phosphate group to the glucose structure. That's what have happened, glucose to glucose 6-phosphate. So we have these 6 carbon atoms, right? These 6 carbon atoms. So this is your core structure of glucose. In the 6th position, 6th carbon position, we have a phosphate here. So this is glucose 6-phosphate. So glucose getting converted into glucose 6-phosphate to make it stable. Okay, now my second question is, where does this phosphate group coming and attaching to the carbon? There is something known as ATP, adenosine triphosphate. ATP constitutes 3 phosphate groups within it. So ATP is being acted by an enzyme called hexokinase. So this hexokinase enzyme will act upon circulating ATP and takes a phosphate group and give it to the glucose. And therefore, the ATP is getting converted into ADP, adenosine diphosphate, 2 phosphates. Where is the other phosphate going? It is going and attaching to the glucose molecule. So that's why how this glucose 6-phosphate is formed. Coming to the point of enzyme again, hexokinase. Remember dear students, kinase is an enzyme which have the capacity to give a phosphate group or to remove a phosphate group. That's about your hexokinase. So this is all about your first step where glucose is getting converted into glucose 6-phosphate under the enzyme called hexokinase. Now, glucose 6-phosphate is getting converted into fructose 6-phosphate. It is fructose 6-phosphate. Glucose and fructose are isomers of each other, meaning the chemical composition remains the same. Even for fructose, it is C6, H12 and O6. All that differs between glucose and fructose is the chemical arrangement. So with the same chemical molecular composition, alteration in the structural organization gives the difference between glucose and fructose. So here, glucose 6-phosphate is getting converted into fructose 6-phosphate in the presence of an enzyme called isomerase. So here, the enzyme is isomerase. Now what happens? Fructose 6-phosphate is getting converted into fructose 1,6-bisphosphate. What is fructose 1,6-bisphosphate? Now we will try to understand. So we have the same carbon core of 6 carbon atoms, and we are having 6th position. We are already having the phosphate group. Now in addition, we have this first carbon atom with the phosphate group. So we have seen now there is one more phosphate group which came and stuck to the first carbon atom. Where does this phosphate group coming from? Same way, there is circulating ATP which is getting converted into ADP and giving one phosphate group getting attached to the first carbon atom. So for this to happen, we also need an enzyme, right? What was that enzyme? Again, I said you it will be a kinase undoubtedly. But what kinase? Phosphofructokinase. It's phosphofructokinase which acts upon the circulating ATP and takes away the phosphate group and give it to the first carbon atom, releasing ADP, adenosine diphosphate. Now, look at the linear structure of fructose 1,6-bis is 2 away from each other. 1,6-bisphosphate. It's a long linear structure which is unstable and breaks down at this juncture, with 3 carbon atoms forming one group and 3 carbon atoms forming another group. Let me name those groups now. So this particular group 1, 2 and 3, and first carbon atom we are having a phosphate group. Again 1, 2 and 3, the second set. Third carbon atom is having the phosphate group. Now, the third carbon atom with the phosphate group is known as glyceraldehyde 3-phosphate. It is known as glyceraldehyde 3-phosphate. And 3 carbon atoms with the first position phosphate group is known as dihydroxyacetone phosphate. Dihydroxyacetone phosphate. So, now fructose 1,6-bisphosphate is splitting into 2 groups which are known as glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. For this split to happen, we need an enzyme known as aldolase. It's aldolase which is the enzyme which is splitting fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Always remember, this dihydroxyacetone phosphate have every chance to get converted into glyceraldehyde 3-phosphate. This is a reversible reaction. Even this is a reversible reaction. Glucose 6-phosphate to fructose 6-phosphate is also a reversible reaction, and this is also a reversible reaction. So now, one molecule of glyceraldehyde 3-phosphate will be there and another molecule of dihydroxyacetone phosphate gets converted into glyceraldehyde 3-phosphate. With an enzyme here, for this conversion we need an enzyme which is known as triose phosphate isomerase. Okay, for the conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, we need an enzyme called triose phosphate isomerase. So here we have 2 molecules of glyceraldehyde 3-phosphate. Let us try to understand this glyceraldehyde 3-phosphate will get converted into 1,3-bisphosphoglycerate. Let me draw the chemical structure of 1,3-bisphosphoglycerate. So we have 3 carbon atoms, right? It got split. So we have 3 carbon atoms. In the first place we have a phosphate group 1. First place we have a phosphate group. And second place we also have a phosphate. Third carbon atom we have a phosphate group. So 1 first carbon, 3 third carbon, bisphosphate, phosphoglycerate. So this glyceraldehyde 3-phosphate is getting converted into 1,3-bisphosphoglycerate. Now, before I explain you from where does this phosphate group have come and stuck to the carbon, there is a beautiful intermediary reaction which takes place at this point. That is conversion of NAD plus into NADH. Now, what is this NAD plus? NAD plus is a molecule which having lot of hunger to accept, to steal, to draw the electrons from the other molecules. So this NAD plus have the capacity to draw the electrons. Now, where does these electrons coming from? Hydrogen atoms which are present in glyceraldehyde 3-phosphate structure. So, from the glyceraldehyde 3-phosphate, NAD plus will take 2 molecules of hydrogen. Let me tell you, 2 molecules of hydrogen. Hydrogen will contain 1 proton and 1 electron. So, first hydrogen what NAD plus takes will take up complete proton and electron. Okay. Then from the second hydrogen molecule it takes up only the electron, leaving behind the proton freely. That's why NAD plus is getting converted into NADH with 2 electrons and 1 proton in this NADH and leaving behind the extra proton from the second hydrogen atom. Okay, so there are 2 hydrogen atoms taken up by 1 NAD plus. So in these 2 hydrogen atoms, first hydrogen it takes up both electrons and protons. In the second hydrogen it takes up only electron, leaving the H plus. So that's why it's NADH and H plus. So this is what is important to understand, because this NADH and H plus is rich of electrons. Those electrons are very important in electron transport chain that we are going to discuss later in lectures, to release the energy, to release the ATP. So that's the importance of NAD plus converting into NADH and H plus. Now, our previous question, where does this phosphate group coming from? It's nothing like here what is happening. It is not like ATP is donating the phosphate, but directly it is the addition of inorganic phosphate. It is the addition of inorganic phosphate. So there is an inorganic phosphate which gets and stuck to the first position of the carbon atom and making it 1,3-bisphosphoglycerate. Now, for this reaction to happen, again we need an enzyme. What is that enzyme? Glyceraldehyde 3-phosphate is this particular molecule, glyceraldehyde 3-phosphate. We want to remove the hydrogen and give to NAD plus, right? So that's why it is dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. I think you all understood why it is dehydrogenase. Because from glyceraldehyde 3-phosphate we are removing the hydrogen, stealing the hydrogens and giving to NAD plus, and there it's getting converted into NADH and H plus. So the enzyme here is glyceraldehyde 3-phosphate dehydrogenase. Now, this 1,3-bisphosphoglycerate is getting converted into 3-phosphoglycerate. How come this phosphate group is kicked out? I said you the kinase enzyme. This kinase enzyme, phosphoglycerate kinase. So this kinase enzyme has the capacity to remove the phosphate also. What it will do after removing? We have circulating ADP, diphosphate. So this phosphoglycerate kinase will remove this phosphate and give to this ADP so that we can see it getting converted into triphosphate, adenosine triphosphate, ATP. That's how the ADP is getting converted into ATP and the energy molecule is released by the action of phosphoglycerate kinase. Now this 3-phosphoglycerate is getting converted into 2-phosphoglycerate. So this 2-phosphoglycerate, there is only the change in the phosphate group from third position to second position. That's it. That's why this is phosphoglycerate mutase is the enzyme. Mutase. Phosphoglycerate mutase is the enzyme. This 2-phosphoglycerate is now converted into phosphoenol pyruvate. So what is phosphoenol pyruvate? How is this getting converted? It's in presence of enolase, an enzyme called enolase. Now, this 2-phosphoglycerate is getting converted into phosphoenol pyruvate. What is that? 3 carbon atoms with the second carbon atom having an oxygen molecule. Attached to it is your phosphate. And there is a double bond between your second and third carbon atom. That is the chemical structure of phosphoenol pyruvate. Let me take you back for a while, dear students. Remember that there are 2 molecules of glyceraldehyde 3-phosphate, 2 molecules of NAD+. As I have said, this is one is direct glyceraldehyde 3-phosphate, 1 molecule, and the other molecule of G3P is your dihydroxyacetone phosphate getting converted into glyceraldehyde 3-phosphate. So 2 molecules of glyceraldehyde 3-phosphate, 2 molecules of NAD, 2 molecules of NADH and 2 molecules of H+, 2 molecules of glyceraldehyde 3-phosphate, 2 molecules of 3-phosphoglycerate, 2 molecules of 2-phosphoglycerate, 2 molecules of phosphoenol pyruvate. Now, this phosphoenol pyruvate is finally getting converted into pyruvate in the presence of pyruvate kinase. So again, here this phosphate group which is remaining is kicked out by an enzyme called pyruvate kinase and giving rise to the end product pyruvate. This kicked out phosphate in the phosphoenol pyruvate is given or donated to ADP, and then here also after addition of this phosphate group it is getting converted into ATP, triose phosphate, and by removing all the phosphate groups there is a new product which is known as pyruvate, which has the end product of glycolysis. This is how the glucose is getting converted into pyruvate. Now, in the next Krebs cycle which happens in mitochondria, we will try to understand how these 2 molecules, remember 1 molecule of glucose is giving 2 molecules of pyruvate. From here, there are 2 molecules of glyceraldehyde 3-phosphate giving rise to the 2 molecules of pyruvate. So 1 molecule of glucose will give rise to 2 molecules of pyruvate. So let us try to understand how this pyruvate gives rise to lots of energy in mitochondria in the upcoming lecture on Krebs cycle. Thank you so much for watching.